recognized extensively for his efforts. In particular, this year, Nuno received the Gilbert Ahmed Computational Proteomics Award of the US Human Proteome Organization. And just very recently, he is this year the recipient of the Beeman Medal of the American Society for Mass Spectrometry. And we are very fortunate to have a chance to hear from Nuno today. And welcome, Nuno. Thank you very much for being part of my institute.
Thanks, Olga, for the very nice introduction and for the invitation to be here. This is really a prime forum for talking about mass spectrometry and talking about the sorts of things that we do. And it really is my pleasure to be a part of this program and to be able to talk to all of you here today about the sorts of things we're doing with regards to, helping enable reproducibility, help promote reproducible analysis of data. And in connection to this topic today, now talking about it from the perspective of the data and how do we make the data available and how do we help share the analyses that were done at the time so that you can then, learn as much as you can, not just about the results that are presented, but also about the way in which the analyses were done. So in the context of this topic, I'll start by giving a bit of an idea of where we're coming from with Massive. And then I'll talk about the Massive Quant effort that we put together to go more in this direction of having reproducible research, not just at the ID level, but also at the level of quant, which obviously is critical for the analysis of biological data. So you might have seen this from the outside. Know that there is a proteome exchange consortium that is now pretty well known in terms of data deposition, that when papers are published, data has to go somewhere that is independent of the researchers that derive those data sets. And this was started about 10 years ago. And you can see that this slide, which I keep at this point for historical reasons, in terms of how things were in 2016, 5 years ago, that we can see then in contrast to that, what does it look like today? So 5 years ago, we had 4,500 public data sets, and it was a quest to get it to that point when we started. Things moved very slowly. But since then, as you can see, things have changed very quickly. So in 2019, when we presented Massive Quant effort for the first time, things were close to about twice as many data sets as originally deposited. And today, in the last 2 years, it's already almost doubled again to, almost 3 times, almost 4 times as many data sets as 5 years ago. So things are growing really quickly and data is becoming available really quickly. So the question we want to consider is, how do we make the most of this data? How do we make it possible to release this data in a way that can be most informative to support future research, as well as to explain the research that was originally associated with those data sets? So in general, the view that, a fair number of people have of how repositories work is that you do your work, and at the end, right before submitting your paper, the journal has that nagging guideline and you just have to put it somewhere in a repository and it just goes up to stay there. And then, maybe a couple of people later down the road might be able to download that. But at Massive, we wanted to challenge this view because as it is today, because of the way that data is deposited, most of the time you actually have a pretty challenging task if we want to reuse anything of the data that was deposited, because data sets are deposited as raw data and the analysis results, if any, are deposited in a proprietary format. And that usually makes it complicated for others to open and to be able to see what was done in those analyses. So, the picture, unfortunately, looks sort of like this in general, that only, 1 in 20 data sets roughly has reusable identifications in the sense that they're released in an open format that you can open with tools, open source tools, and most of them essentially have no reusable quant data. If deposited, it's in a format that you can't easily look at. So Massive from the beginning was designed to restructure the data and to help it be reanalyzed, to be converted to open formats whenever we can. We try to convert the formats that are submitted by the users into open standard formats that you can then open in other tools and use standard ways to analyze those data. The data can be automatically reanalyzed, and we have done quite a sure amount of reanalysis, and we'll talk a little bit about it today. And then we want to provide advanced visualization interfaces that you can use to analyze the data without having to download it. You're very welcome to download it if you'd like, but since we're talking about hundreds of terabytes of data, it is preferable if there are ways to help you interact with those large volumes of data without having to download it, because it's just too much to download. With the goal being that hopefully this can help integrate this data into your research process, rather than being something that comes only after the whole research is finished and you're just needing to get an identifier to submit your publication to a journal. So there are a few things that you'll see when you come to the Massive homepage, and the most common two things are shown right at the top of that page. And that is, if you want to browse the data sets that are available and search them by metadata, that's one of the options that's available at the top. And the other one is to submit your data. Those are the most common operations that we see. But I wanted to also mention what it is like to have a data set when it's submitted to Massive. And there are a couple of things that you see on a data set page when you get to it, because the most important point here is that a data set is not static. It is one thing when it is first submitted, but it can and does evolve over time. So the first thing is you get the identifiers for a data set. And in this case, we see that this is a data set that was actually originally released in the NCI, or National Cancer Institute, Proteomic Data Commons. And then it was imported into the Proteome Exchange Consortium with this Massive identifier, and thus assign that Proteome Exchange identifier. So you can find data sets that are present in multiple repositories. Definitely, even if data was originally deposited to PRIDE, or in this case, to the Proteomics Data Commons, it can still be imported into Massive for reanalysis and reintegration into these tools. And in particular, for reanalysis with Massive Quant, which is the topic that we'll get to here today. So we do provide the standard operations that you expect to see on data sets. You can add or update metadata on your data set. You can add publications to a data set. We also provide the ability to add files to data sets. Not all repositories let you do that, but if you need to replace a file in your original data set, you're able to do that. You cannot delete things that are in data sets. Data sets are immutable. So once published, then they can't be actually once submitted, not just once published, because during the review process, it also should not change. So once a data set is created, you can't delete anything from it, but you can add things to it. But we also allow the other kinds of options that allow you to expand on the things that you can do with the data set. So in particular, you can convert the spectra to open formats, which is an important step that is necessary for you to reanalyze it with open source software. So this can be done online for you. You don't have to download and do that. And once the spectra are converted, then you can reanalyze it with a variety of different tools. There are, your standard database search tools, library search tools, proteogenomic search tools, open modification search tools, so on and so forth. You can upload your own. If you did an offline analysis, you can also upload your own and attach it to a data set. And we'll hear more today about how to do this for quantitative results. But you can also run max quant and MS stats directly on the system, which is something that has been a great interaction we've had with Olga's group to put together this ability to do the quantitative analysis of the mass spec data directly online without having to download the data and figure out just how to do everything offline. You can actually do these analyses online now also.
No,
And
there's a
final,
question. Can we change the name of submitted files?
I'm sorry. Where was the question?
If
I
it's
had the
possible to,
answered open,
yeah.
not the open ones. I'm sorry. Thank you for reminding me of that. Can we change the names of submitted files? Interesting question. Actually, interesting as it sounds, I don't think it's ever come up. Generally speaking, we wouldn't, but I can imagine cases where it does make sense. So it doesn't actually change the file. So I would venture to say yes. I think that's an edge case that we hadn't considered. But I know that in some cases, the metadata files have some file names that differ from the file names on the data set. And definitely corrections like that would be welcome. So I would go ahead and say yes, because that does not change the raw data. And that is really the intent of this guideline for the whole Proteome Exchange Consortium, is that the data itself does not change. But if you're just changing the file names, presumably to make it clearer what the data set submission is, then I would think that makes sense to allow to do that. It would have to be something that we would have to do. It would have to be an interaction that happens over email with us, so the system doesn't provide that option online, but we would find a way to do it, to especially if it is to clarify the purpose and the content of the data set. Absolutely, I think it would make sense. Thank you for the question. So I was going from the ways in which we can reanalyze, and then these reanalyses are added on the system. And in this case, for this particular data set we're looking at, there's already a series of reanalyses of different kinds of tools and with different kinds of results, and they all appear attached to the data set that as was originally submitted. So you don't have to go to a different place. As long as you can see the page for the data set, you'll see all the reanalyses that have happened for the data set. And then what can you do with it? So there are tools provided in the system that allow you to compare reanalyses. And that helps because you may want to compare different tools and you want to see how the gains and the sensitivity of the different tools compare. You can compare different protocols. You can compare different samples to see if you're finding the same proteins or not the same proteins in different data sets. And the way that this works, you can select results that are attached to data sets, or you can select any of your private searches to compare to those data set results. And then you get results at the spectrum, peptide, and protein levels, where you can see how the two sets of results compare. Now, if you want to go beyond the pairwise analysis and comparison of results, you can actually search all the identifications that are submitted in Massive. And as it is today, it's originally it was over 300 million, and technically it's still over 300 million, but now it's over 500 million identifications that are available in a system from dozens of terabytes of data. And you can use those to look for any protein that you're interested in or any peptide that you're interested in. You can find all the data sets in which it occurs, and you can find all the modification versions in which a peptide occurs, and so on and so forth. So it gives you the ability to search across all of the data in the system. And if you want to use a distilled version of all these searches to help with new searches, then we have the Massive KB, which stands for Massive Knowledge Base Spectral Library, that you can download with over 2 million precursors of reference spectra. And there's a protein explorer interface that allows you to see all of this information in a protein-centric manner, instead of just being more data-centric, where you're looking at data sets and you're looking at the data set's metadata, or the spectrum-centric view of the spectral libraries. Here it's a protein-centric access into the system, where you come with your protein of interest and you can see all the information that's available for that protein. So it is in this context that we come to massive quant, in the understanding that quantification obviously is one of the most important, if not the most important, kind of analysis that you can make on proteomics data sets. And we really needed to have a way to represent the complexity of the protocols that are used for the analysis of this data. And as you can see, just briefly illustrated here on this slide, there's very many different ways in which you can combine tools for identification and tools for quantification to then do the statistical analysis to get the results, the quantitative results and the differential abundance results that you then use to understand the biology in the samples that the data sets come from. So there were many different ways in which these protocols happen, and we see here different studies that combined these different ways of analyzing the data in different ways. And each path that you see from the top to the bottom are different ways in which the data sets at the top were analyzed to result in different reanalyses. So unfortunately, we don't even have standard formats for all of these. There are some standard formats for ID, but for quantification, that is actually something that is even still being discussed, as well as for the metadata that would be necessary to describe the samples and to give you a chance to run the quantitative analysis. You need to know which files go with which samples and which conditions so that you can then do the analysis. So the approach that we took for massive quant was to try to come up with a way to capture all of this information so that we can begin to share these studies in a meaningful way that has enough information to understand how the studies were actually conducted. So with this goal in mind, we teamed up with Olga and Meena and put together the Massive Quant resource, which builds on Massive's ability to store data sets and to represent data sets, and adds the additional things that we need to represent quantitative data sets, where we aim to capture the metadata that's necessary for these analyses, as well as the scripts and the results that are derived along the way, as well as the final results, of course, of the quantitative analysis. So as it is today, we are at slightly over 200 reanalyses that are quantitative and give you differential abundance results of 64 data sets across a variety of different to interacting with all of you to see how we can facilitate the sharing of your analyses and how we can make these results more useful for you. So it's great to be here today and have the opportunity to have that conversation. So there's a question here of whether this is limited to DDA, and the answer is no, it is not. The Massive Quant resource can take results from any kind of quantitative proteomics experiment. In particular, it can also take DIA results or targeted analysis results. As long as the quantitative tables are available, then we can have that be the quantitative information that's made available for the data set or for the reanalysis, and include that as the result, as the quantitative table. Now, we wouldn't have tools to process that kind of data. So there are some tools in Massive to process DIA data, But note that you don't have to use any of our tools. You could have done all of your analysis offline and then just submit the files that you got from your offline analysis. If you wish to use some of the tools that are available online to assist with the analysis, then at this point, they are more DDA centric. So for the both label-free and isobaric labeling experiments in DDA mode, we can actually support the full analysis being done online, either on public data or on your private data. But for the other kinds of data types, such as DIA and targeted, Massive Quant still supports it as a submission, but we don't have the tools to do it online. But it's still supported and definitely welcome, because at the end of the day, it is the differential abundance results and the proteomics results that are most important, and we'd definitely be glad to have them. So some of these reanalyses here already are of DIA data sets and of targeted data. So we could definitely highlight those as examples that could be used to start new submissions if you're interested in going in that direction. So the sets of new features that had to be developed for this system essentially fell into three major categories. The first one being we needed to have a new data set format. There's another question here. Let's see. Staggered DI data, okay, for massive, is it better submit non-demultiplexed raw or converted plus demultiplexed MZML? Great question. So I would actually recommend submit both, that you can submit both the raw files as well as the deconvoluted and converted, demultiplexed in this case, MZMLs. Both would be welcome. And in some cases, we have DIA data analyzed with DIA umpire. And that's an example where we have both. We would have both the original swath files as well as the deconvoluted MS2 files. And there's actually more than one. It outputs more than one. All of those can be submitted in separate collections. So when a data set is submitted, there are different subsets of the data. There's a collection for raw data, and then there's a collection for converted files in open formats or standard formats. And that's where the converted and demultiplexed data would go, with the raw data going in the raw collection. But both would be the best, just to make sure that we have the best record of what was originally done. Because as you know, there's different ways to convert the data. Sometimes there's centroiding, sometimes there isn't. And there's definitely different ways to demultiplex DIA data. So the best way to make sure we have a record of what was done would be to have both of them in there. The other question is it possible to retrieve a large set of proteins peptides to generate a spectral library with massive tools? If so, are there limitations? So let me see, two different ways to answer this. The first one being, if we're talking about human, we already have a spectral library that covers a lot of the data that is available out there. And it has actually, I mentioned earlier in the talk that it was 2 million precursors. It has actually been expanded recently, and it's now almost at 4 million precursors. So for human, we have a pretty comprehensive spectral library already in place. Now, if we're not talking about human, or we're talking about a situation where you want a smaller spectral library for a specific kind of tissue or for a specific kind of sample, then there are workflows in the system that allow you to generate those spectral libraries from any set of data sets for which we have reanalysis results. And if we don't have the reanalysis results, then you can, then those can be obtained. Then there are workflows to get those reanalysis results. So it is possible with, it is possible in both cases. So if it is using the whole human library that's already available to download, and it's a simple step to just essentially download it. And if you want to make new libraries for either different species or for different specific conditions, then it is also possible to do that, because the exact same workflows that we use to build our libraries are publicly available for anyone to make other libraries. And essentially with the difference being what the input data is for those other libraries. Are there limitations? I'm sure there are. I don't want to say that there's low limitations and anything is possible, but I can tell you that in terms of volume, our human libraries, start with dozens of terabytes of data, and they start from, hundreds of millions of PSMs. So the system is able to scale to those levels. I guess what I would say in terms of limitations at this point is you would need to start from DDA data. And right now it is expecting either CID or HCD type of spectra. There are some filters that have to do with explain intensity. So if you're using ETD or ETHCD or different kinds of fragmentation modes, the workflows currently are not equipped to deal with that. So I would say, as long as you stay within DDA and you stay within CID, HCD, high res or low res is fine, then you should be able to build those. So great questions, everyone. Let me then go back here and say that in terms of the dataset formats, what we needed to add to the infrastructure we already had to support raw data and open data and reanalysis, identifications reanalysis, we wanted to be able to support the kind of data that's important for the quantitative analysis. So we wanted to be able to capture the metadata for those data sets. And because, for reason one, there was no standard format to represent that. And reason two, we want to make this an easy next step for people who are already doing the quantitative analysis. We actually made this metadata format be modeled after the MSStats input format. So the same file that you learned how to configure here, or you already knew from before, it contains all the critical information necessary to describe how the data, the files that you have in the data set or in your study, relate to the samples and replicates in your experimental design. So to make things easier, we just essentially adopted that format and said, if you can submit that metadata, then you can make a quantitative data set or reanalysis submission. The other two collections contain the quantification results. These would be either the peptide abundances or protein abundances that you get from your quantitative analysis. Could also be TMT ratios or other isobaric labeling ratios. And then there's a final collection for statistics that contains the statistical analysis of the quantitative data. And that is where you would see whether things are differentially abundant or not. And if they are, at what level of significance. That's essentially the distinction between these two. One of them is the quantification results. The other one is fold variations and statistical significance of those fold variations. And then finally, as I already alluded a little bit earlier, we also wanted to provide executable workflows that you can use directly in the system to run the analysis, to facilitate running your own analysis, as well as the analysis of public data. So these are workflows that you can use for both private and public data. So if you upload your data to Massive, you can actually work with it and analyze it in the process of your research project, and none of it is public. You actually have to explicitly go through a series of steps to make anything public. So you'll never make anything public by mistake. And it is ideally the better way to go and do things if the tools are available in the system. We'd be glad to support that research process directly online. And one of the reasons I really like to do it online, one is that it makes it easier for me, that I don't have to write things on a command line. And if I'm doing it for many reanalyses, it's easy to just clone a job and rerun the analysis. But one other reason is that these online tools also provide you with interactive results views that show you in this particular case, the results of the MS stats analysis. You can see all the differential abundance results. And if you click on the little graphic icon for each row, you get to see how the different abundances split against the different conditions, and you get to see the box plots for the different comparisons that were done for the particular analyte in that row, which in this case is the protein level analytes. So one last point I wanted to make here is that a recent batch of reanalysis that has been included in Massive Quant derived from efforts in this past year for the analysis of SARS-CoV-2 and COVID-19 data sets, as well as data sets of related viruses. And these cover a variety of different experimental types, as well as different types of omics. There's also metabolomics and lipidomics data included in this Corona MassKB resource. Reanalysis including PTMs and ID, but most importantly in the context here today, also quantitative reanalysis, including MS stats results that are now available and can be accessed through either system. You can access it through the Corona MassKB resource, or you can access it through the Massive Quant resource. So with that, I'll just stop here and let give us a moment to ponder this high-level view of what the system does. And then Meena can tell us a little bit more about how specifically you can submit reanalysis into the system and interact with the system for either submission of data sets or reanalysis. If we use the executable workflows online, can the results be shared with collaborators online? Absolutely. It's actually very simple to do that. When you get your results, there is a link that has a random string in it. And if you just copy paste that link and email it to anyone, they can see the results that you derived. And you don't have to remember that link, because in your account, we keep track of all the jobs that you ran. And if you click on that list, you can see all your past jobs. So you don't have to go and memorize or save it. But yes, it's very easy to share results with anyone online. In the online analysis of the workflows are somehow optimized? You mean automated or authorized? Whereas users...
Automatic.
Automatic.
Yeah.
Yes. So there are definitely, as users can work with the tools offline like we do, online like we do offline, choosing all the different parameters. Yes, so, okay, yes, maybe, depends on the parameters. For most tools, there is an input form in which you can select values from many parameters. But for a tool that is as sophisticated as MS Stats, there's always going to be a bit of a trade-off. So if your study is not a standard type of study, if it's a less common kind of study, then there may be things that could be done offline that can't be done online. But for most studies, in particular case control types of studies, multiple group types of studies, those sorts of things you can definitely configure and run online. And you can select things, you can select filters like minimum number of peptides per protein, minimum number of bioreplicates per group. There are things like that that you can configure in the input forms for the tools. So the goal is to be able to capture maybe 90% of all the analysis that you can do with those tools. But we do recognize that the 100% is not possible, because in some cases, you need some specialized types of pre-processing of the data that currently may not be supported. But in general, I would imagine that the majority of cases would be supported. Now, before transitioning, I just wanted to mention something very quickly that unfortunately I need to teach at the same time as this session. But it pains me, because I really like the fact that we are here interacting with you and we can hear your questions. So please, if there's anything that I'm not able to answer here and you'd like to consider or is something that is in your mind before deciding whether to move forward and try the system, please do make sure to contact me or contact us if needed, to keep the conversation going. Um, so quickly, there's another question here. Is it possible to implement massive tools locally? So the workflows are made available as open source. However, there's a number of things that are done at Massive that are based on accessing public data. So from that perspective, it's kind of intertwined with the system. But when it comes to the algorithms, we definitely make an effort to make those workflows available to run locally if you'd prefer to do it that way. Or to look at the source code, absolutely. We do make an effort to release them all as open source.
Nuno, thank you very much for the
Thank you.
presentation and thank you joining us. We know your teaching schedule was changed at the last minute, so that's what happened. We are very grateful that you could make it regardless of that. And indeed, for everyone, please don't hesitate to reach out to Nuno with more detailed questions after this session. But at this point, let me welcome back Meena. So as you have heard from the previous session, so Meena is currently a research scientist at Genentech, working on all things quantitative mass spectrometry. And before joining Genentech, Meena was the lead developer of MS Stats. But in addition to that, Meena also was the leader of this effort that integrated the workflows in MS Stats with the data and workflows in Massive Quant. And so for the rest of the time today, we will talk about some specific details of how we can use quantitative workflows within Massive Quant. Thank you very much, Meena.
Thank you, Olga. So we had a really great session this morning. Talk about the reproducibility, what they learn, and then the whole pictures of the massive and massive quanta from Nuno. So the goal for this section is I want to introduce the massive and the massive quant a little bit more detail. So all the aspect of what the Nuno mentioned, I will just show the briefly and then give you the all the sources you can follow later. Um, and then I will not use the present mode because I just want to go back and forth, back and forth. But if you have a difficulty to read my screen, please let me know. Okay, so massive is look like this. So let's go to the massive first. So I think that you can get the slide, and then this is the link. You can go to the massive. This is the first page. So here's the massive one. If we scroll down, you can see there's many things that we can do it. We can access the public data set. We can submit the data. We can search identification, and so on. And then the bottom, we can see here's the massive quant. So here, if you click the link, Massive Quant, this is how the Massive Quant web page. At the beginning, there's a summary of statistics of the data set. So for example, here we have a 64 public data set, which include the metadata and then quantification data. And then there are more than 200 quant reanalysis data set is there. And then among that data set, the number of the data set by species, so there are 36 data set is for experiment with the human, and then others is the mice and something else. And then if you scroll down, there's a list of the item what we can do in the massive quant. So we can browse the public data set. We can submit the reanalysis. We can access the reanalysis. And then we can use the MS workflow in the massive quant. And then we can compare the workflow. We can compare the MS that output in the massive quant and so on. So each section you can click. So for example, if you want to browse the public data set, you can click this, or you can click the documentation. So there's each step, each task, we have a documentation link. And also we have a video tutorial. So if you wanna the summary analysis, you can check the documentation. Let's click this, how it look like. There's all the documentation. You can just follow one by one. So here, so upload the data and then click next. You have to add the analysis and then something else. So there's tons of the documentation there. Also, if you click the video tutorials, we have a video. I will just stop doing playing. But in the Ogavitex lab, there are, so for example, this one is for 15. So I will just record my screenshot to how to, you can submit the reanalysis. So you can just follow each step over there, but I will go over shortly today. So first step. So for massive quant, what we can do, we can just browse the public data and submit the analysis. The browse of public data is quite important for like the computational person like me, because sometimes we want to play around the data set from publication. So they submit the data somewhere, but we want to access this one, right? So, or we want to searching. I want a human data set for specific acquisition type, and then actually we can search this. So this is the goal for the browse the public data. Also, some reanalysis means we don't need a road, we don't need a new experiment. We just want to use the public data experiment, but we want to reanalyze that data, and then we want to submit it. So to share it, like just for multiple purpose, maybe that is for maybe also another computation or the paper to propose, or if we want to share this one, or we want to compare in the different other result in the message quant. Also, we can access the reanalysis, which means we can access the reanalysis from others. And then we can also just look at how they're doing and etc. Also, we can do the MS-DEC workflow in the message quant and then compare it. And then there's a lot of the documentation there. Then let's see how we can browse the public data set. So here's the message quant. And then let's click the browser data set. So this is the how it look like. So massive quantity data, there's a big tables to show, to list the whole the data set which require, which is satisfy the requirement for the massive quant here. So you can see whole the list of the tables, right? And how we can make your data set available in the massive quant is if you have the quantification data result, means quantification data, and then you have a study design, means another metadata, what are gonna talk to you soon, and then the result of differential abundance result. If you submit 3 data set, it will gonna automatically show up in the message quant. So for example, here, total 64 data set already submit this data, and then the message quant to recognize this data set, have this information, and then here it is. So for example, here, this data, I have a keyword. This one is the DI data. And then for red plasma, for the specific the research question. And then they have this one. And then from metadata, they automatically count this experiment have 17 different conditions. And then they have 42 biological replicate there. And then each one they have up to 5 technical replicate. And then they have 279 proteins are quantified, and then among them 231 is a differential abundance of proteins. So, and then you can also see who is the PI, and then what kind of instrument we use, etc. And then here's another column called the reanalysis container. So I'm going to talk about what is this means, but which means in this data set, there are several reanalysis there or not. So for example, first one, they don't have it, but second one, they have another space to store the reanalysis. Sometimes they have a plus sign, means they have a multiple container to save more number of the reanalysis there and so on. So this is how it looked like. I also want to show this one. So this table is a filter by any data set with the quantification result and then differential abundance, real analysis, and the study design. There are also, we have a review level. So the bronze, silver, gold, the platinum, and so on. So let's just click the platinum. So there are 25 platinum one, which means they have all the information, and then we confirm we actually can reproduce the result. So which means they have a fully documented data, and then we need all the requirement information to reproduce their result. So this is the how you also can use this one. So then maybe I want to look at the select data set what we play around last a couple of weeks. So yeah, let's see where we have this data, or I maybe they have a next page. For example, here TMT10 control mixture is the data set the team play around yesterday. So you can also go there and then see all the output from there. Here we go. So this data set is the what we play around. So I will click this one. So here, title SWAS data set from East over the time in the response to osmotic stress. So here's the description there, right? And then here, they also have a summary of all the data set in this the data set. And then you can see also we have a publication. They have owner, which means the first the submitter of for the raw files. Raw files, the owner did not provide experiment design one or quantification data. However, later we submit the 22 reanalysis. So we can just also double check this one. And then bottom, you can see also the list of the whole reanalysis analysis. So we're gonna look at this one a little bit and then just also play around on what we can do it. So cell-based analysis, we can say the two things. One is submit the new data, new experiment, and then we can submit the reanalysis. The new data means we are going to submit the raw data, start from the raw data, and then we are going to submit the intermediate. You can submit the intermediate one. Submit the reanalysis means raw files are somewhere there. So data set have their own unique massive ID. So this ID is assigned for each experiment, each data set. And then this data set says this is the public, so you can get the public data set. And also we have the proteome exchange, the ID as well. So if you already submit your data set in the proteome exchange, we actually, the massive also can import to your data if we have this ID, and then we can also, you can have also another ID for massive. So if you already have this MSID include the raw file, and then I just want to reanalysis, and then we want to submit it. To, if you just go back to the holder step for data analysis, this is a kind of the brief step we can do it, right? Though we can do the sample preparation, and then we got the data, which I will call the primitive raw files. And then we have to do the spectral identification and then quantification, and then we can do the statistical analysis. Last couple of days, we talk about how we can do this statistical analysis step using the quantification data, right? And before, we also talk about how we can do identification and quantification using Skyline. This is what also we talk about last week and last week, right? Here is some tools or the file, like intermediate file input or output for each step. So for example, sample preparation, we actually don't have it, but after we got go through the sample to mass spec instrument, first we're gonna have a raw file, right? And then do the identification, we might need the signal database, right? So this is also should be input for identification or quantification step, right? So raw file or FASTA file or something else will be just input for this identification step. So here's one of the example. If we use the max quad, so we just put this information in there, and then max quad output quite a lot of the intermediate file, right? Among them, the MQPAR XML or parameter txt is the parameter file, which record what kind of parameter we used in the version numbers and so on for the max quant. And then also one of the output we are going to use for statistical analysis, the evidence file or group protein group file. So we call this is the quantification file, right? To use the MSTER or MSTER TMT, we need this quantification file. And then we probably use the R script if we use the R in locals. And then annotation, the CSV, we call this is another way the metadata, which means metadata include your experiment design. So up to here, we it's not a big deal to each raw file from which human sample or their age or their group, right? But statistical analysis step, it is important. This, each row corresponding to what kind of sample we have to use this one. Example of the annotation. This is also what we practiced with the team yesterday. This information is the required. So for example, here each run means each MS run. Each sample is corresponding to what kind of condition and then from which biological replicate, right? For TMT cases, it will be, we have probably the one more column called channel, right? Because each channel have a different sample information. So this is the we call the annotation, and then based on this, we count how many conditions we have, and then how many biological replicate we have, and how many technical replicate we have, and then also we also count how many runs we have, and etc. So to get the final the statistical analysis, to get the same, if we want to reproduce the same the result, actually we need the whole intermediate file, right? If you're missing one of them, actually we cannot get this one. So for example, if we missing one of the quantification data, definitely we cannot get this one, right? And also parameter file is also very important. If we have a different version of the other tools, maybe something is changes, we cannot get the same result, right? If you have a different parameter setting, definitely we are going to have a different output. So to get full the reproducibility for this our final output, all this information is needed, and then we can save all the intermediate file into the massive quant. Then this is the for one analysis, one reanalysis. But each step we can use the several different things, right? So for example, we have a one experiment, and then we have a one set of the raw file. But maybe we can use the different the searching and identification algorithm. And after that, we also might use the different processing tool to get the quantification. And then for statistical analysis part, we can use the MSStat. However, we can use also different the statistical method. For example, we talk about the bioconductor today, right? So if we go to the bioconductor, this one is to hold the storage to all different type of the R of the package, to give you the idea how you can find some other the R package available there or something. Here's a use, and then if you click the software, this one lists up the whole the package available in the bioconductor. So for example, if you click the software, if you click the research field, I will say research field, there's a proteomics. So there are 145 is our package related to proteomics one. And then this one is linked by number of the download and so on. So for example, Lima, also we can use the Lima for proteomics one. And then Lawrence, the package, yeah, Lawrence, we can see the Lawrence, the package quite often here, right? So you can see whole the R package, and there are many different way to you can analyze it, right? So each step, each based on each your decision for each step, output will be different. So, but we want to save this one more systematic way in the one place and easy to access it. We have a question. So in case we are using another processing tool, so we are Skyline Spectral Mine, should we provide the software output in the MSD friendly format, and or we run the no. So the goal is we don't want to lean on the very specific software. So that's why we want to make everything so generalize it, right? So this is the one of the case just to show you the demo. So if you use the max quant, this is the output, right? But if you use the Skyline, Skyline doesn't report the evidence file. Skyline doesn't report the protein groups. So Skyline report the MS3 for the format, right? That will be the quantification, and then we can use it. They probably also report some different parameter file. Then we can use a different one. Spectral Mine also they have a different type of the quantification, and then also sometimes, so for example, Skyline they have a Skyline specific format dot SKY or Spectranon dot SNE or Proteome Discoverer .pd. That is also output from their tools, right? We can also save it as a quantification widget, and then we can just bring back. So this is the kind of the one of the idea, but at each intermediate file format or file type might be different. You don't need to run everything through the max quant, but it is required to submit the intermediate file to get the final output. So as much as you can, for example, log file, parameter file, it is very important to record your reanalysis. Right. So this is the whole idea. So we want to save all your selection in each step. So how the messy quant structuralize it is a massive quant support the branch structures. So at the top, the high level, we are start from the data set. It could be one experiment or one publication. And then massive quant to give the ID start MSV number 000 something like this. So this is the ID for the specific the data set. Under the data set, we can make a several container, right? So under the data set, we can save a container, and then this container have also new ID start with the RMSV. So for example, I just want to analyze that specific data is already available in there. I will make the my container there, right? Or maybe Ting later, Ting also do the reanalysis later, and then she can make a container, her own container under the data set, right? So we have a two separate container. And each container, maybe I want to try the several different parameter setting to analyze the data set, right? Then each parameter setting, we have a different analysis. Then we have another unique ID for the reanalysis, which is RMSV dot number dot X. So we have a branch structure data set, and then we can make multiple container there. And then each the container we can make a reanalysis. And each reanalysis we have a one space kind of the folder, and then we can put all the intermediate file there. And then we have an ID and link. So whenever you want, you can just give this link to others, or we can just save this link for the future purpose. So here is the one of the example. So this is the one of the data set, and I just make a two container. And then for first container, I reanalyze a set of the raw files using different processing tools. So first of all, this reanalysis processed by Skyline. And then another reanalysis is processed by MaxQuant. And then another one we processed by the Progenesis. And then also with a different setting. So we can, from the one set of the raw file, there are 10 different reanalysis. I can save it up there. And then I also make another container, how I can use the MSDAT the parameter. The first one is the different processing tools and their parameter setting. Second container, I've made how I can use the different parameter setting for MSDAT. But how you can organize this is quite up to you. It's a kind of how you organize the folder. But the important thing is the message quant to have a support this branch structure. You can save all the output as much as you can. Also, I saw some other cases is data set is for one publication. But sometimes one publication, we have a several different set of the experiment, right? It's up to you. You can make each experiment, you can make a new massive ID, or to submit the paper or to share the data set, make a data set for publication level. And then in container, we can submit each experiment. In that cases, we have a different set of the raw file for in our container, right? But the data set level, they're gonna contain the whole set of the raw file there. But in the container, we can select which raw file we use for this specific analysis and so on. So, okay, so this is how you can organize it. But to submit the reanalyze, how we can do it, and then what we needed. To submit the new experiment, there's the documentation there. I'm going to show that briefly. To add the reanalysis to massive quant in the existing data set, three things are required. One is the quantification data. For example, MSDAT input, which is a Skyline output, what we practice, which means the feature level data. This is a starting point. I want to use this one for statistical analysis. Metadata is exactly what we show. It should include the information of your experiment design for each run from which samples. So this is the quite simple cases, but and then we use the East one. However, if you have a human data set and age, gender, some other really good important information we use, definitely you can add this information in the column. But the three column is the required. And Then the last one is the output of differential abundance analysis. Definitely we need the log to 4 changes, and then p-value or just p-value column is required for this one. If you use some different statistical analysis, definitely you can submit the output in here, and then you can fully document it in the Reanalysis, the branch. Other thing is the parameter setting file is not requirement. However, I strongly recommend to submit this additional information. Also, the R script or log file to record how you use the R or Python and so on. Definitely, it would be good to submit it there. Okay, so you have all the set of the data set, and then I'm ready to submit it. And then we never used the message one before. What should we do is the 3 steps. First, you need to register the massive account. And then you have to put all the file in local computer to the massive space. Then you can do it. So here's the video and then documentation, but I will gonna show you how to do this. So when we go to the massive page, you can see here top right the user and then sign. So I already made my account, so I can just click the sign in. But if you don't have it registered, this is the only information needed. So no extra step. Just to put save your user name and the email, password, and submit, and that's it. So you can start to use it. So I will sign it, and then please use the login to use our workflow. The successful login, right? So now here we go. To submit the new data set, submit new data, your data. And then there's nothing here, but it will take a few seconds to us show whole things. Here, the workflow selection. The workflow is what the Nuno said today. Here is already selected the massive data set submission workflow. So we can just start from there if you want to submit the new data. However, if you click the button, here's the whole list of the workflow what the message want to support. So for example, here you can add the file, convert. You can use the MS-DEC workflow in there. MS-DEC comparison workflow, we can use it. Add comment, add Reanalysis, pride import, right? So if you know the pride ID, you can just import your data set into the massive. So this is the workflow and etc. Right? So to submit the new data set here, and then you need to give them. So here's a data set metadata, means experiment third part. So species, instrument, who is the PI, and then description, and so on. So as I mentioned, whole documentation is there. Let's see some or one of the document there. Right, so here's as what we showed there. So really detailed step there. You can just follow it, how you can select the species and so on, right? One thing I want to point out the submission file category. So we have a several different types of files. Someone ask about the MGML, MDID, what kind of information we needed for ID part. So here's the list. So for example, require for complete submit, you need a peak list of file, and then this is the file type, and then reject to files, and then requirement for all submission, which means this is really required. If we don't have a raw file, we cannot set up the submit a new experiment, right? And then also searching engine file. And then also the optional one. So here is the optional one, and so on. So I recommend to check this part if you want to submit a new one. And then, okay, so now we have a account for massive. And then let's go to the massive quant, right? And then I want to submit the reanalysis. To submit the reanalysis, we have to know the massive ID first, the ID for existing data set you submitted. And also we have to um, load the files what we got from local computer to the massive space. In that case, there are several ways to upload it. There's a documentation there, but I use the FileZilla, the FTP uh, one. So here is the FileZilla. So here you have to type massive ucsd.edu, and then you have to use the massive quant ID you generated, and then password, and then connect. This is okay, right? Then we have a laptop panel and then right panel. The laptop panel is my local, my computer. The right panel is a space of your account in the massive. So we have to transfer file to the right side, right? If you are the first time to use the message, probably it would be empty. But I put the many data set already there, so it looked like a lot. But it's the same as what you can play around. So for example, I want to submit the select data set what we practice last couple of days to the massive quant one. So here's the MySpace. So you can just organize as much as you can. Just recommend to name it um, that can help you to easy to find it. So for example, here you can right click and then create the directory, right? You can create the directory as you what we want. Um, Yesterday I made the new folder because it will take time to like around 1 GB to the massive. So I made it this one. So here is the left panel is the my folder, what we practice. So here's the output folder, what we practice. So low CV and then output there, right? I need the MSTAT input plus one, which will be the quantification result. And then our script, what we practice, it will be good evidence to how we can get it. And then this is the test result, which include the log for changes and then so on. And I should upload the metadata about the experiment design. So how I did it is just click it and then just move to the right, and then this is already uploaded there. Yeah, so here's already successfully transferred. And then this ms.log file is also the generate from the mstat. This also include all the parameter what we use for mstat, so it will be good to save it, right? This one actually have a big file, so it will take a time. If you have a raw file, if you want to make a new submission, it will also take a while because it will be like several gigabytes per each raw file. So please have the good the internet connection. If it's disconnected, maybe you have to start from again. But now it is done. So now all the files, what I want to submit it is already in the massive space. Then I will go to the web page and then I'm gonna call this one in the using the interface. Then here is the Celebstick data set. Well, so I will sign it. After I sign it, there are several more buttons that show up, right? So before I don't have it, but here's the add reanalysis one, and then add file. So add file means not about the reanalysis. At the real top level data set, I want to add the file over there. And then also upload the metadata, which means contact information or keyword and so on. Also add the publication. I already add the publication here. You can also add your publication after um, your manuscript is published. So here, click the Add Reanalysis button. Then here you can see workflow, add reanalysis result is selected. And then you can title for that specific reanalysis. So I will say May Institute Sergeant 1 Massive Quant Hands-on, right? There are some limit of number of the characters or required number of the character. We can see if something wrong, they're gonna show up what's wrong. And then the metadata is already selected from our top level data set. So this experiment used the EAST1, used this instrument, and so on. So you don't need to worry about it, but if you want, you can add it as well. Another requirement is we need to add description about this specific reanalysis. So we have to put something here. What is this reanalysis for? So I said, this is the result using the MSD during the May Institute 2020 week 2 hands-on sessions, right? So here we put the title, we put the data, the descriptions, and then here is we need to select the file, which file we want to add for this analysis. So here, for example, peak list and row spectra files already selected because this one automatically selected from containers. So then I want to submit the quantification result, etc. So you can click one of this. So everything link to the one position here. So new window is pop up, right? Then here is the whole list of the data set what I interact with. But bottom, you can see your massive ID. And then click open. And then this one show your this right side. This one show you your space, your account space in the massive. Then I made this self-sec folder, right? And then I made a new directory under there, right? And then I submit this one. So I want to submit this file. You can see the files you uploading use the FTP one. There are several files, but you need to tell them which file for which output. So for example, this one, MSD input plus the CSV is output of the Skyline report. And then this one is the input for MSD, right? Then if you click, you can see this is a highlighted, like light blue, right? If you click again, it's gone, right? So highlighted light blue means select this. I select this and then go up here. This one is quantification result. Then under the quantification result section, this file is selected. And then, okay, the R script, I selected. And then which section? R script is a method. The method section, we can put it in the method section. So this one is now is under the method and protocol sections, right? and then come back again. Annotation is about experiment design. On the metadata, here we have to click this one is some metadata. If you are not sure which file should be which section, so documentation have an example there. So and then show the what kind of information we needed. So you can also check this documentation there. Test result is statistical result, right? Statistical analysis of the quantified analysis. So you can click this one here. And then last, MSDAT and session info, you can just check both of them. And then also you should go to the method and then protocol. So we select all of them, right? Yeah, we select all of them. And then the raw file, I didn't select it, but if you submit the new one, select the raw files and then click the raw spectrum file, and then you're gonna submit it here. And then I'm done, the finish selections, right? And then you can see here the quantification result, the one file selected, method, and the 3 files selected, and etc. Right? Then here, next step is you can assign the container. So I want to submit this reanalysis under which container. So here, let's click this. So there are 2 containers there, right? So you can submit one of this, or you can make a new analysis container. So let's make a new analysis container. And then I will say here is the May Institute 2021. And then you can also define the password. And this part, you don't need to worry about it for the analysis part. And then here's a show the email me at here. And then if something happened for this workflow, they're gonna send you the notification. This workflow is done or fail, something like that. If you fail, definitely we have to come back and then check it. So then we can just click submit, right? Right here's the error creating test. This one required upload the peak list is not there. So then, okay, come back. Yeah, and then you can double check this one. Let's say we are not selected this one. Let's remove it, right? And see how it goes. Right. So now massive quant is running to set up the pipeline, the container structure, and then find the location. So it will show up the progress. So it will show the what rows spectra file is linked to this analysis, and then you can see this. At this moment, you don't need to worry about it. Maybe it will take some minutes, or it's a really big file. It will take some time, but whenever it's done, you can see. Yeah, here's how it show the progress, how it works. Another thing is here in the massive job, it show the workflow what you tried. For example, here click, it show the what I tried. So sometimes I fail, sometimes it's done, and then the last task of what I try. This is the running. So this is the how you can submit the reanalysis one. Then while we are waiting for, let's try to look at the what we already submitted, how it look like. If we succeed, how it should look like, right? So let's go back to our SelfSec data set. Here we go. So now at the bottom, you can see hold the title and reanalysis container. So I have a question. To everyone with the account can share new reanalysis? Yes, if this submitting process is successfully done. If fail, it's not, but if successfully done, yes, you can see it. So if you made a count, definitely you can see this or workflow. So this one I will show you. So there are 22. I will go back to the very first one. So this is the one I submitted 2 years ago, the same data set with the some setting, right? And then this is the container. So this is the data set, and then this is the container. And under the container, we have the real analysis. So let's click this. So this, I just put a little bit detail description how I analyzed this data set using Skyline. So here's the using Skyline daily version, blah, blah, blah. And then this one is used for this publication and so on, right? And then show this reanalysis used 11, have a total 11 files, this gigabyte. Each one have a 6 conditions. And there are around 900 proteins are significantly differently or differently abundant. And then total number of the proteins to quantify are more than 2000, right? And then if you go to the FTP, you just download this link, you can directly download it. However, there are also some other buttons there. Let's just see how it looks like. So browse the reanalysis file. I will click this. So if you click this, it will show the whole the files under this reanalysis. So for this reanalysis, you can see there are the input is there under the quant directory, right? And then also they have a Skyline G file too. This is it, this is under the others because there's no specific folder for tool specific files, right? So, but you can download this diff file and then you can open it in the Skyline too. And then also R script there to generate this one, and then annotation files, some parameter files as well, and then testing one. So for example, let's click the annotation file here. This is probably smaller. Here's a size 2. Click it, and then you can download it, right? Or here up there, there's a filter by collections, right? So if you click the quant file, it show under the quant file, this file you can see, you can also download it. QuantStat, you also show the QuantStat file, which is the output of statistical analysis. You can download it, etc. QuantStat others method have no restriction about the file types. You can upload it any file. So you can upload a PDF, JPG, anything you want. You can upload it this one. This is the how we can browse the reanalysis. So I want to go back. It's still running. Yes. And also, this is already published, and then this is imported from Pride, right? And now this is the public, so it's already public one. But if you want to submit a data set for publication, prefers want to keep it a private before you accept your publication accepted, definitely there's an option. You can just make everything is a private, which means other people cannot see, only you can see that data set. And then also can have you can make ID and password. You can share the reviewers. So, and after everything is going well, and then after done, there's also make a button to make public. And then if you click it, after everything is done, this one will be show up to everybody. So then you can actually share this link. One good thing is if you submit everything is here, in the publication, there are data availability sections, or sometimes you have to fill in some other document. You can give them just this link, and then they can just see all the files under here. We have out of time, so that is the most important part. But I will just show you just other things about the MSTEP workflow there. The concept for MSDAT workflow is currently, let's just come back to our data analysis workflow, right? So we have a raw file over there, and then from the raw file to the final testing output, we still need a several step, right? What we commonly do is we have a public data set there. Then if you want to rerun the identification or quantification, we have to download to the locker, right? Download the locker and then do the identification or quantification or something. And then we just need to upload all the file up there, right? And then run it. However, now this workflow, if we have a data set available under the here, we can use the pipeline, the workflow in the massive quant, and then automatically save the output under the right the categories. Um, let's go back to our first page in the massive. Here, there's no button to use the MSDAC workflow. Still, just key is to just click the submit your data set, and then we can actually select the MSDAC workflow here. So first, it looked like this one, but if you click here, MSDAT, then give the massive some second. Yes, so now the page look different, right? So we can type the different title so you can recognize it, and then you can select the files, or you can say what kind of tool you use. This is the list of the Spectra data processing tools MSDAT can support. So basically, the MSDAT have a converter for them or not. So we have a converter for this of this, right? So we can actually use them. So if you want to use the max quant, which means now the massive quant say what kind of input we needed, right? And then the label free or TMT or iTrack. So if you click the TMT, it will gonna call the MSDAT TMT, not the MSDAT, and etc. Then you can also select the files, which is important to run the MS test. Definitely we need the metadata, the annotation files, and then we need the features or proteins. So same as before, click the input, and then you can select your input file here, something like this. Or if you already submit this one under the data set, you can select this button. And then you have to searching your the massive ID. I forgot what is the ID. Let's see here. 8. Okay, let's copy this here, and then click filter. Yeah, it will show up, right? And then let's go to reanalysis, and they show the whole reanalysis there, right? And then the hands-on sections. Hands-on. So now you can see this one. Let's say this one is should be annotation, and then I add it. And then the MSDAT, this one should be feature level data, right? And edit and close it, right? So there's a question, what if my experiment type is the SciLec, right? So SciLec, technically there's no MSDAT SciLec one. So we can use the MSDAT TMT for SciLec, but we have to be careful how to make a annotation. So maybe we have to go back to the tutorial what the team covered yesterday. But yes, for SciLec, you can use the TMT. Right, so select this, then you can set up the comparison one. So kind of the one versus other and etc. So all versus all means all pairwise comparison. Or if you want to click the one means all pairwise against one specific groups. And also you can choose it, or you can actually make your own contrast metrics, and then you can input it. Here's the advanced filtering. This is the option. Some of the options not cover the whole MSDAT one, but if you click here, there are some options there. So for example, you want to input yes or no, right? what kind of normalization one. This is the default, but I don't want to, or I want the quantile. If something like this, and then we used all feature, but I want to use the only top end, or I want to use the high quality, is based on our feature selection algorithm, and then click submit it. Then same as the what we practice, it will show up this one. Okay, so this one is done, right? So if this is the done, then let's go back to our um, this site, right? So now 22, let's um, now we have a 23. So here we go. Now we can see our reanalysis submitted in the message quant. Quickly, the other one. So I use the same MStat output. So now every reanalysis have a same format under there. So then actually the message quant to compare our testing result. So this is the how we can do it the same as MStat workflow. You can choose the MStat comparison one, and then select which analysis, which output you want to compare. If the output format is different, probably have errors. So we should be careful about the output of the testing you want to compare. Last, in our slide, there's a lot of the link for documentation. So please revisit if you want to submit your data and the reanalysis. And then also there are the videos for each step. So you are stuck for one specific step, please just go there. And then it has more details. So now we have not many times, but it will show all the detail step one by step. So that is all. If you have a question, please let me know.
Thank you so much, Meena, and thank you to everyone who stayed all the way through the end. I think this is a really very important contribution to be able to document all the analysis, not just related to the identification of spectra, but all the way through statistical analysis of quantitative data, and do this in a way that is not specific to a particular tool for data processing or particular statistical analysis software package or anything like that. So even if you are a method developer and develop your own statistical methods or your own data processing methods, this is a repository for you. And also if you are working more on the biology or on the clinical side, and you're interested in documenting your work or just look at the results of the analysis of similar data sets, we also hope that this will be useful for you. So with that, this concludes our session today. However, we will stay back as always